join me in welcoming this evening's speaker, Professor Alain Goriely.
Thank you very much, Richard. Thank you, everybody, for coming, and whoever is online, hello. So today we will continue our mathematical exploration of the brain. In the first two chapters, we discuss the effect of size on brain performance, the effect of geometry. But today, we will look at the wiring, at the connection within the brain, what we call in mathematics the topology of the brain. So what do I mean by that? If I look at the brain, this beautiful slice of a brain, what I see is these long filaments, transporting information between different regions of the brain. What they are axons. Axon is part of the most important cell in the brain, which are neurons. And they have this long process connecting one neuron body to another neuron, transporting information. These cells are responsible for all the cognitive function and for all sensation and control of motion and so on. And what we see here is this long filament within the brain that connect different parts. Now, I can identify different parts, so let me circle some of them, different regions that I know may be associated with memory or with motor control and all that. And I can trace these filaments. They are actually huge bundles of filaments, maybe hundreds or thousands of these filaments together, and see how they connect one part of the brain to the other part of the brain. And this is what I see, different type of connection. Not all parts of the brain are connected. For instance, I don't see any connection between the frontal cortex and the visual cortex. It has to go through other parts of the brain. And so there is structure there. So forget a moment about the brain and just let's try to extract this structure. In mathematics, such a structure is called a graph or network. We'll use the word network today. And what is a network? In the most simplest term, it's a set of nodes connected by edges. That's all there is. And what we want is to extract information from this structure, the topology, are they related, and be able to say something meaningful about the organization of the brain. And the particular neural network of the brain is so important that it has now a name that is in the Oxford English Dictionary. It's called the connectome, which is like the genome, the connection of the neural network. There are plenty of networks. It's not just the connectome. You also find other networks almost everywhere where you look. If you think at different entities that are in relation, you'll find a network underlying this. Think of a family tree. In this case, the nodes are individual, and you have an edge if they're related by blood or by marriage. That's a simple one. What about the World Wide Web? This is a big network. Here it's in infancy. Before it became the internet, it was a baby internet. It was called the ARPANET in 1969. It just had 13 nodes and 16 edges. By now, it has grown, or baby is fully grown. It has 1 billion websites with maybe 100 to 1 trillion connection. So 1 billion nodes, 1 trillion edges in this huge graph. Another one that we hear probably too much of it are all the neural networks that are under everything we hear about AI and ChatGPT and large language model and so on, every day in the news. So these have a structure, neural network, artificial neural network, that's actually quite different than the human brain. And here is an example. The idea is that you take information in, for instance, the pixel on a photograph, and then you have all these layers that are connected, and they call them neurons. And all edges connecting layers by layers, transforming the information until at the end you have an output that tells you it's either a cat or a dog, or what is the next words in the language. Of course, this is a very simple architecture, the one I show. The one that it's actually used in large language model is based on that, but it's much more complicated. It's the so-called transformer. And to give you an example how big these are, GPT-3 has about 2.4 million neurons and 175 billion edges. By now, GPT-4 has tens of millions of these nodes and maybe trillions of parameters that encode a lot of our human knowledge, or at least the human knowledge that is in text. Here is a more interesting or more ludic network, characters in a novel. I show you here the different character that appears in Lord of the Ring, and these are nodes. Each character is a node. And I connect an edge if they appear in the same paragraph within the text. And right away, you see some structure appearing from that network. You see that there are characters that really dominate the whole text, like Gandalf and so on, and Aragorn and blah, blah, blah. All this great. And you see right away that just by doing this simple exercise of connecting things, I can see the structure, underlying structure that is in the novel. Important, industrially important example are the electric grid. Here is a map of the UK, where each node is a station of a substation, and the edge are actual physical electric line connecting these. And again, that structure architecture is very important if you want resilience, redundance, and all that. You have to think about where to put this line and are they connected and what station do you need. In particular, if there is a problem in one of these, you want to know how it cascades through the whole network. Very important network. Other ones that you're familiar with is social media. If you think that people who have an account, that's one node, and you have an edge if you're friend with somebody or if you post on their account, you can have different type of network. And of course, this is very important for all modern relation. Yet another one is the air transportation network. And this, for instance, I'll show you with 3,200 airports, is a very big network that's very important, and I'll come back to that in a second. But we are here to talk about the brain, and we want to study the network of the brain, the connectome. So we need some. So how are we going to get that? So let me show you 3 types of connectome that we study in science: worms, flies, and men. And I start with worms in this 1937 Mickey Mouse cartoon, where Mickey Mouse actually plays the role of a mad scientist that transforms worms. Very worth watching. You can go on YouTube and enjoy it. The worm in question is a little worm, a round worm. It's about 2 millimeters long, and it was chosen around 1963 by a very well-known biologist, Sid Brenner, because it was so simple to study. It's transparent, it's easy to reproduce, it has a life cycle of 3.5 days, and he really wanted to understand the genomic development, organ regulation, and so on. And he studied these organisms. He chose his organisms for this purpose. It became so successful, he eventually got the Nobel Prize in 2002 for his research. And the same organism has given people 8 different scientists a Nobel Prize. It became so important. The most difficult part of this organism is its name. It's Cynoarbaidis elegans. But because it's so difficult, it's known to all its friends, and all the biologists in the world as just C. elegans. So whenever we talk about it, it's just C. elegans, because that makes it much simpler. So the idea that Sid Brenner has, besides studying the complete genome and see the effect on making genetic experiment, is to study the connectome. It says it's simple enough. We can look at all of these individual neurons and try to trace them and see how they're connected to each other. Try to extract the connectome. And so he started with his students to try to do that. And it's very difficult. Imagine that you have a big sausage, this worm, which is actually 2 millimeters, where you have a sausage, you can slice it, and in each slide you will see the neuron, the axon passing through, and that would make just a little dot like the one you see. And then you have to identify on the next slide where is the same dot, the same neuron, and so on. It took them 10 painful years in order to do that by hand. They tried with computer, they programmed. Computers were not good enough at that time. But the result was very impressive. In 1986, they published the first complete connectome, the first full neural network, in a paper published in the Philosophical Transaction of the Royal Society of London, which is 450 pages long, with all detailed map of that. What does it look like? This is the connectome of the C. elegans. It has 302 neurons and 7,725 edges. And you see, it's looks complicated, but it's the simplest one that we know everything about. Just to tell you the kind of things we can extract from that, here is a little portion of the connectome, and in brown and blue you see the forward and backward premotor and interneurons. So these are particular neurons that are tasked to initiate or modulate motor output, either going forward or backward for the worm. And so if you can target them, if you can control them, you can control the motion remotely of the worm. And that actually has been done recently, or relatively recently. Very nice experiment where you can do some laser ablation of this neuron and control either forward or backward motion. So we know extremely well the behavior of this and how it's related to neuron. That's the first one. And you see it's relatively simple, 300 neurons, not that many. And for many years, we did not have any other complete connectome until the fly. And you see here the fly represented by this 1958 horror movie that you might remember. 1986 was the remake of the movie that you might have seen with Jeff Goldblum. Not great movie. Story of a mad scientist that by mistake match or mix his own DNA with the DNA of a fly and transform during the movie. It doesn't end well for all the people involved, of course. So, 1986, movie remake of The Fly. 1986, C. elegans, full connectome. It took until 2024 to do the full connectome of the fly, the fruit fly, which is another model organism that is extremely well studied in biology. 1986 to almost 40 years it took, because it's so complicated. And here is the fly in question, Drosophila melanogaster, is, again, for his friend, it's just called Drosophila. It's very well known. We know just about every possible aspect of his genome, about his behavior and all that. Wonderful experiment. But now we have the full connectome. So what do we have? Of course, you saw on the poster the big eye. Here is the head of the fly. And what we know is what's inside the head now. We know exactly what's inside. And we know that this brain of the fly, this nervous system, is composed of 78 different compartments, each involving different type of computation that support behavior such as sight, flight, memory, and so on. And if you peel that, now we have the complete map of how it's connected. And it has 139,000 nodes and 54 million edges. And you really needed not only the power of modern computer, but also the development of AI supervised by biologists and humans in order to be able to do that. Huge progress, and it's only 4 months old. It's really happening now, the kind of knowledge that we have. Let me show you 2 neurons within these. And these are the 2 neurons I want to show you. These are called the CT1 neurons, the giant neurons. So the filament in blue is connected to the eye, you see, with all these little pieces. And these are 2 neurons, one on the left and one on the right, that are tasked to sample the amount of light in order for navigation to happen for the fly. And here is the full 3-dimensional detail of these 2 neurons. Absolutely beautiful. Neurons come in all different shapes and form and lengths, and these are particularly large, but you could have very small neurons also, or very long, like the one that connect the brain of a giraffe all the way to its leg. It's also single neurons, or the one on a blue whale that might be 20 or 30 meters long, right? You have all possibilities when it comes to neurons. Wonderful diversity of shape and function. Here is another set of neurons, this type only 4 neurons, and that they create a pathway for thermosensory. We know so well about this. So this is what explains the ability of a fly to sample the temperature and fly away from it, which if the temperature is too high. So we know a lot, and we know soon more because we have the full map. So we have C. elegans, we have the fly, great. What about the human brain? Here is poster from a 1936 British science fiction, also horror film, where Boris Karloff plays, as you can guess, a mad scientist who develops a technique to transfer minds between bodies. And it doesn't turn out well, as you can guess. So what about the human brain? Well, if you remember, in chapter 1, we studied the size of the brain, and we established that there are about between 60 and 95 billion neurons in the brain. So we are nowhere close to be able to have the full connectome. We need a different approach altogether. Maybe at some point we'll be able to have a full connectome, but that's science fiction for the time being. It's way too complicated. The best we can do is the 100,000 neurons for the fly at this point in time. So what is the approach? It says, well, we can't do the whole thing. We have to think differently. We have to go back to the geometry of the brain, and for instance, to this theory of Karl Brodmann that the brain is more or less divided into different regions. They call region of interest by neuroscientists. And in this case, Brodmann says there are 52 that believe that they serve unique functional purpose. Again, memory or motor control or something like that. And the idea is that since we know this, we can try to see how these really reduced sets of regions are connected to each other. We don't know the full thing, but we might know the macroscopic picture of this connection, and maybe we can learn something out of it. So let's try to do that, and to do that, I'll have the help of my colleague, Johannes Weikenmeier. He was a postdoc in Stanford at the time when we studied his brain. He's now a professor in Oxford and works in brain trauma and aging, and we'll have time to talk about that in a few lectures about his work also. So we put Johannes in a scanner, and what we did is a form of magnetic resonance imaging called diffusion tensor imaging. So MRI, magnetic resonance imaging, allows you to essentially trace molecules of water. So if you take 2 snapshots in time of molecules of water, you can see the motion of these molecules. It turns out that you can also show that the motion is mostly along axon or axonal bundle. And so by tracking the motion of water, you can track the geometry of axons locally. And the picture that you get of Johannes's brain is something like that. What you see is this line where we know the trajectory of water at different places. Here is different views, sagittal view, and so on. And we can now look at his brain, and we can say, okay, let's look at his region of interest. Neuroscientists know there are different types of classification they call atlas, and they can have maybe 50 like Brodmann, but today it might be 80, all the way to 1 million of different regions where we know the different region in the brain. So now what we do is we say, okay, I know the position of the region of interest, and these are going to be my nodes. And since I know the bundles through these imaging techniques going from one place to the other one, I can trace edges where I have an edge if 2 parts of the brains are connected. Not all parts of the brains are connected. For instance, the top of the brain on the left and the right, the frontal pole, it's called, are not connected to each other. There is no direct connection between these two, as you can see. But you can start building a connectome like that. And we've done it for his brain, but we've done it also for a big database that exists out of 400 brains. This is not Johannes's brain. This is an average of 400 different brains represented on an atlas with 83 nodes. Okay, so now we have connectomes. We have this graph. We can represent them in space on the computer. How do we extract something meaningful about it? How do we learn from the graph? And to do that, we're going to talk about networks and matrices. You can already guess that there is a movie theme underlying here. Okay, network and matrices. So what is a network? I give you first a definition. A network, you need 2 elements for the network, the set of nodes and the set of edge. I told you, very simple structure. There is no trick here. There's no big math. The central object is very simple. Let me give you a very simple graph that we're going to use to build on our intuition. Here is a graph with 6 nodes. So the set of nodes is 1, 2, 3, 4, 5, 6. And what is the set of edge? Is whenever 2 nodes are connected, I'll put that in my collection of edges. For instance, 1 is connected to 2, So I put that 1 and 2 in my set of edge, my collection of edge. 1 to 5 is also connected and so on. So I have all the collection. Instead of a picture, now I have an algebraic way to code the edges and the nodes. It's not the best way. There are much more powerful way. And now, starting with that, we're going to build something else to also code the graph. And that is called the adjacency matrix, hence the reference to matrices. And what is the adjacency matrix? First, let me show you, and then I'll tell you how it's built. Here, if I look at node 1, I see node 1 is connected to node 2. So in row 1 and column 2, I'm going to put a 0. 1 is not connected to itself, so I put 0. 1 is connected to 2, so it's a 1. 1 is not connected to 3, so that's 0, and so on. 2 is connected to 1. So in row 2, column 1, I also place a 1. Right? And so now I have these two. I have this edge. I know one is connected to the other, and the other one is connected. I make this difference between the two because I can also have directed network where things go only one way. Information can go one way and not the other way. But we only deal with undirected network. So if 1 is connected to 2 today, 2 is also connected to 1. What about 1 and 5? Well, row 1, column 5, row 5, column 1, I have a 1. I have 4 connected to 5. That's over here. 2 connected to 3, over there. 4 connected to 6, over there. 5 to 2, 4 to 3, and I think I have the entire network here, the only place where I have 1. Okay, it's just a table of number. We all agree that if I give you the matrix, you can draw the graph, and if I give you the graph, you can get the matrix, right? You can go set 1, 1, 2, okay? We all good? So now, why do we do that? What's so special about this one? Well, it turns out that we're very good at manipulating matrices. Not only we are good, but computers are very good at that. Essentially, what computers are very good at, apart from logical operation, is doing multiplication and sums, right? And that is what we do with matrices. This big large language model, what they do is addition and multiplication of matrices one way or the other at a very fast pace. And so it's very easy to extract information when you have a matrix. Okay, so what about the connectome now? What is the matrix for the connectome? What is the adjacency matrix? Well, I can label my node, 1, 2, 3, 4, 5, up to 83 in this case. And here is the connectome, where you have a black dot if you have a connection, and you have it's white if you don't have any connection. So now I have a matrix that's 83 by 83, and it has this structure. Here is a slightly better version, the color version of the same adjacency matrix. And it has a little bit more information, because I told you between two regions, not only I know if there is a connection, but I can also look how thick is the connection, essentially how many neurons go from one place to the other one, right? So if you think in pipe or if you think in airplanes, you not only want to know if two airports are connected, but how many planes, how many people go from one to the other one. And so you can put that in color, where you have, if you have no fiber, no connection, is going to be in blue, and many fiber is going to be in red. And so this is what the weighted connectome look like, or this adjacency matrix. That with 83 nodes, and this is another one I made for you. It's 1,015 nodes with the graph of the brain going with it. And I think a lot of you have played with matrices in school and do some addition or something like that, extract information, but you've probably never seen a matrix that's related to the brain. And what's amazing with that is that this matrix has directly visual information about our own brain. What do I mean by that? Let's have a look at this matrix. What do we see? We see these two huge blocks first on the diagonal, and very little connection on the other side. And what it tells you is that, as we know, the human brain is separated mostly in two hemispheres that do not talk much to each other, right? So just looking at the matrix where there is color tells you about the division of labor within the human brain. And there is a connection. Most of the connection between the two goes to the corpus callosum, which has a lot of white matter axonal tract in between. And these are the place here that connects the right hemisphere to the left hemisphere, right? And these are this bundle of axon connecting the two. So this matrix, just looking at the matrix, tell you something about our brain, which I'm still amazed to this day. There is more structure that we want to extract. If you look at the diagonal, you see a lot of color. And the diagonal is arranged, the nodes are arranged when they close together. It tells you that nodes that are close together are also very highly wired together. And we really want to try to understand that structure or separation, long communication, and all that. This is what we're going to do. This is the human brain. We can do the same for the fly connectome. This is the adjacency matrix with the fly connectome, with all the different parts. It's slightly different, you see. There is much more connection. The central part is not simply divided in two. There is process in the central part and all that. That tells you that the underlying topology of the fly is actually different than the one of the human brain. It's not a surprise, but it's by itself interesting. Okay, so now we have connectome. We can represent them in matrices. What we want is to learn something about how do we go from one point to another one on the matrix. We want to find path within the graph. And so let's talk about path. And to help us, I'll start with a simple problem from Good Will Hunting. So Good Will Hunting, I don't know if you've seen the movie, it was the breakthrough movie for Matt Damon and Ben Affleck, where Matt Damon plays the titular character, Will Hunting, who is a young janitor at MIT who turns out to be a genius in just about everything. And early in the movie, demonstrates his intellect by answering a very tough math question given to the PhD student of the math professor there. And this is the board. And you see that the board, the first question, here is a graph, it says find the adjacency matrix. Aha, we know how to do that with as good as Will Hunting already, right? We've done that. We know how to go from a graph to an adjacency matrix. Now let's look at the next problem. It says, what is the matrix giving the number of three-step walk? Okay, so it's if I have a graph, it says, oh, where can I go? How many paths is there going from one node to the next one? Okay, so that's an interesting question. So let's look at it in our case, our little network. The question is, how many ways are there to go between two nodes in k steps? That's the general problem, but let's look at it in very simple. Take two nodes, 2 and 4. How many ways are there to go in one step between 2 and 4? None. How many ways are there to go? And it's already in the matrix. If you go row 2, column 4, it says 0. There is no edge. How many ways are there to go from node 2 and node 4 in 2 steps? 2. You can go 2, 3, 4 or 2, 5, 4. 2 ways. Okay? So there are 2 paths of length 2 between node 2 and 4. Okay, here's the interesting thing. If I take this matrix and I take the square of the matrix, so you know you can take the square of numbers. You can also take the square of matrix, which is a series of multiplication and addition, very simple. If you take the square of this matrix, whatever it is, very easy to compute. It takes 2 nanoseconds on a computer to do that, which is the time it takes for length to go about 20 centimeters, so almost instantaneous. The square of the adjacency matrix contains the number of paths between the nodes. The number 2 tells you that there are 2 paths between 2 and 4. Okay, so why do I know that? So obviously, you've done it yourself. You could easily guess the number of paths. So now let me ask you a slightly different question. How many paths of length 17 are there between 2 and 4? You have to go and take your eraser and all that and start looking. I go to 2, 1, 1, 2, 2, 5, blah, 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 17 exactly those. So that's 1 path and you go. Or, we know now, you take the 17th power of the matrix, which is this matrix, and you go row 2 and column 4, And you find that there are 32,788 paths of length 17 between node 2 and node 4. Very easy. Now we can extract information from that matrix about the number of paths between nodes. And that's going to be very important. So now we know how to compute number of paths. Let's talk about a slightly related concept, which are hubs and clubs in this very forgettable TV series and OC movies from the 80s. Let's start with the degree of a node. So the degree is very simple and very important. Fundamental notion is essentially the number of neighbors, right? Node 2 is connected to 3 nodes, so it has degree 3, right? And we can extract that from the matrix, so the 3 neighbors. We can extract that by just summing row 2. We have 1 plus 1 plus 1, and that's equals 3. That's the degree. And why degree is important? Because it tells you about connectivity of one given node with the rest of the network. And this is really the basis, the first idea that's behind what made the success of Google in the first place when they introduced the page ranking algorithm, was to rank page about different websites which have the highest degree in some sense. It's a little bit more complicated than that. But the degree is important because it tells you what nodes are the more connected than other ones. So within this context, what are hubs? Well, hubs is exactly what you think when you take a flight and you have to connect to a so-called hub. These are the nodes that have the highest degree. In this case, there are 3 nodes that have degree 3. And what it gives you, these hubs, is really a skeleton of the graph. Here's again my air transportation network. If I just look at the hubs, so the nodes that are highly connected to other nodes, I get this map. And now I have a much clearer picture of the skeleton of air transportation in the world, with London Heathrow right in the middle being the biggest, most connected airport in the world, and all the other ones. And that really tells you something about the overall organization. Okay? One more concept. We have these high-degree nodes, they're called hubs. And there is another interesting notion, which is called rich club. I love the names here. You'll see other names like that. What is a rich club? Where in this network, you see that the red nodes have high degrees, because they have a lot of neighbors, but they're also connected to each other. They form what is called a rich club. And why is it called like that? Well, the name comes from a sad but true structure of human society, where there are few individuals in power who know a lot of people, the employers, the servant, the follower on Twitter, whatever. And they happen to also know each other. As we know, the billionaires are a good pal. And here is the movie, Titanic, where you see these people smoking cigars and deciding the fate of the world. And they are the rich club. They have a lot of employees, a lot of people they know. They play a special role within the network. And these nodes are connected to each other, and they play a special role in the sense that out of this node, you can also identify communities. It's called clustering, things that are sub-part of the network that are not so connected to the other one. In this case, you have like 4 different communities. Okay, so now we have these IDs, hubs, degrees, rich club. Let's go back to the connectome, and let's see what we can learn from that. And in 2011, there was a very nice paper that showed that the connectome has this rich club structure. There are special nodes, special regions in the brain that have a high degree, they're highly connected, and also connected to each other. And it turns out that there are a large portion of short communication that pass through this sub-network of rich club within the brain. And we know that these are associated with either associative function or integrative function of the brain. So you have associative function when you link different type of information. For instance, you recognize a friend's face. In order to do that, you have to combine visual input, element of the face, with memories. So you have to take 2 types of information and integrate them, associate them in order to have that recognition. Our integrative functions, just about the same idea. In this case, you combine multimodal inputs. For instance, if you drive a car, you have to combine visual, motor, and decision-making function. So you have to integrate these. All these different all these different functions, different type of recognition, if you want, are integrated through the rich club. And the rich club is important, but it's also important in the sense that if something happened to the rich club, if there is a damage, it has a disproportionate effect on the network integrity. For instance, shutting down London Heathrow, we know has a huge effect on the overall transportation network. Here is the cat connectome, that's also very well studied, and it has also the rich club structure. In this case, the nodes are put on a circle, and the red connection are the one that connecting this rich club. These rich clubs form also communities, and you can look at all the nodes that are connected in a certain sense to different parts of the rich club. And you find one that are naturally associated with different functions. For instance, strategic executive function, sensory motor, memory, visual, auditory and language, or olfactory function. And so these are different type of function, different type of sensation that are integrated and pushed back in different place in the brain. What about the fly? Where we know now everything about the connectome of the fly, so we can run all these and look in detail. So there is a natural cutoff for the fly. It has at degree 37. And if you look at that, you'll find 40,000 neurons that are part of the rich club. So 30% of all neurons. So if the connectome of the fly was a society, it'd be an extremely democratic society. Not so many billionaires, but more like, Scandinavian countries maybe. This much more diffuse, different organization. Okay, now we have this notion of rich club. We can try to see how different parts of the brain are connected to each other, how different nodes might be connected. If I take two nodes, what is the minimal path between them? And that's we're back to the idea of 6 degrees of separation. So this was a 1993 movie with Will Smith, and it's actually the theory that's used in the movie, or the underlying theme. And the theory is that any people in the world are connected to at most 6 social links. In other words, you can reach anyone, whether a celebrity, politician, farmer in a remote village in Colombia or something, to a chain of 6 acquaintances. I might not know anybody in Colombia, but I have a friend who's Colombian who knows somebody in Bogota, who knows somebody, and so on. And within 5, 6 steps, you can connect everything. We still don't know if it's true in the world, but a lot of networks have this type of property. And going back to the movie, there is a very nice, interesting game that plays on that theme, which is called the 6 Degrees of Kevin Bacon. In this case, the network a network of movies. The nodes are the actors, and they have an edge between them if they play in the same movie, or if they worked on the same movie. And at some point, Kevin Bacon, in an interview, said, he's worked with everybody in Hollywood. And so people say, let's try to see if that's the case. Can we relate them by like that to people, to different people? So for instance, Kevin Bacon and Stephen Fry have not worked together in a movie. However, Benedict Cumberbatch has worked with Stephen Fry in The Hobbit, if you remember. Benedict plays the dragon, Stephen Fry the mayor. And Kevin Bacon and Benedict Cumberbatch has played in Black Mass, 2015 movie, not that great movie with FBI and stuff. So in that case, we say the Bacon number of Benedict Cumberbatch is 1, and the Bacon number of Stephen Fry is 2, because it's 2 degrees of separation. Okay? And, here maybe I stretch it a little, but here is me. Since I work for the movie Sherlock Holmes, and I did the board for them, I can claim that I have, on a good day, I have Bacon number 3. Turns out that 250,000 actors have a Bacon number 2. It tells you how big the connection goes. And the total number of linkable actors, if you look at the Internet Movie Database, about 4 million. And only 1.5 million of them are actually related to Kevin Bacon. So he hasn't worked with everybody in Hollywood or is connected. But you can try to find the average path length in these. What do I mean by the average path length? Is that I can look at the minimal path between two nodes, the minimal number of degree of separation. Okay, so 1 is connected to 5, so the length is 1. The minimal length between 1 and 4 is 2. I go to 5 and 4. The minimal length between 1 and 6 is 3, 1, 5, 4, 6. So I do that. And I can do that between any two nodes. And then I look at the average of that by all the possibility. And that gives me a number, which is the average path length. In this case, it's 5/3. If I do that the same with the Bacon number, with the actor one, I find that the average path length between two actors that are among the 1.5 million is 3. There's 3 degrees of separation. Essentially, if you take a random actor, the chance is that it's going to be 3, right? More or less. It's not the best, by the way. Samuel Jackson has a number, a Bacon number, or Samuel L. Jackson number, actually lower than Kevin Bacon's work with more people or more connected. You can do the same with Facebook and all that. It turns out that people are extremely connected, and the average distance on Facebook and all these social media is somewhere between 3 and 4 in most cases. Okay, so now if we want to design a network that has good communication property, we'll go back to the brain in a second, what we want is this average path length to be small, right? Because we want to pass information, and we don't have to pass information one to the next. We like to be direct communication. So the worst case is to put all the nodes in a line, a very simple network, but it's really bad in terms of communication because the average path length is n+1/3. We could do the computation is not very difficult, but it's an interesting exercise. It's a different number if you put them on a circle, but it's n+1/3. What is important is that it scales as n. If I have a network that's 10 times bigger, the average path length is 10 times longer, 10 times bigger also. So if I go to 1 million, 10 million, 100, it gets longer and longer to pass information because I have to go one node after another. Really the worst possible architecture to design communication process. The best case, of course, is that every node is connected to each other. And in this case, the average length is 1. Okay, so the average path length is really a measure of long-range connection. Okay, that's communication ability within a network. Now, the best case scenario is a little, of course, it's easy. You connect everything to everything. But that's extremely costly. If you have 1 billion nodes, okay, the number of edges on the line is 1 billion minus 1. But the number of edges, if everything is connected, is n times n, is n squared. So 1 billion is 10 to the 9. You need 10 to the 18 edges. That's extremely costly if you want to wire something where everything is connected to it. Okay? So we'll go back to that. How can we design, with a finite number of edges, a good network that has short communication length? Very easy to communicate. We'll go back to that in a second. First, let's do the exercise for the brain that we know. So that's the adjacency matrix. It's not that hard an exercise to do to compute the average path length. It's not completely obvious either, but there are nice algorithms in order to do that. And in this case, for this particular one, the one that I built, the average path length is 1.5. Very short. So this brain, this connectome, is very short path length, very easy to find a connection between two on average. That brings us to a notion how to design a network that has the right property in terms of communication. And that brings us to a very nice concept, which is called small worldness. And there are a bunch of movies, actually, that are called It's a Small World. Here is a 1963 movie called It's a Small World with Spencer Tracy. And now the question is, if I have a finite number of edges, what is the best way to reduce path length, but also allow for this little computation that I need in a brain to occur? What do I mean by communication? If I look at a picture of a square, I need to be able to detect the edge of the square. And that requires local computation. And once I have that, I need to integrate the fact that it has other edges. And so a lot of things are happening in the visual cortex very quickly when I see a square that have to happen at that part. lots of local computation about recognizing different features of an object. Okay, I have to do this computation, and then I have to send that information somewhere else in the brain, says, oh, the name is square, and that's other part, that's part related to words and all that. Okay, so how do we do that? We need yet another concept, which we want the nodes that are close together to be highly connected to each other. And this is the concept of clustering, or clustering coefficient. It's, in a graph, the relative number of connected triangles. What do I mean by that? You have a high clustering if any of your two friends are friends with each other. Very simple concept. That means that you form a little community, right? And if you think that if your two friends know each other, I have one edge to one friend, one edge to another friend, and the two friends know each other, so that makes a third edge. We make a triangle between the three. And so the question now, I can look at a graph and find all the nodes that have two friends, or two neighbors, and see if these friends also are connected to each other. On average, is that big or high? Are all your friends know each other, essentially? So I look at the relative number of connected triangles. In my little graph here, I have 5 nodes that are connected to 2 nodes, but there is only one triangle. Okay, so in that case, the clustering coefficient is 1.5. And what is that important? Because remember, we want to do all this local computation, and so the clustering coefficient is a measure of close connection. The average path length was a measure of long-distance communication or connection. The clustering coefficient is a measure of the close one. And so we come to a very important notion, the notion of small worldness. And that was introduced by my friend Steve Strogatz, and at the time his PhD student Duncan Watts. It's a brilliant piece of work published in 1998, one of the most influential scientific paper that really started all the network studies and all that, hugely successful. But the idea is absolutely beautiful, and the beauty of it is that it's so simple. Okay, so here's what they say. Let's start with a very regular graph. I put all my nodes in a circle, and I connect all the neighbors and next door neighbors. Okay, so they're all connected like that, as you see on the left. And the problem with that is that the good thing is that I have high clustering because all my neighbors are also connected because of this double connection, next door neighbor also connected. So high clustering, but long path length. Why is long path length? I have the same problem as before. If I try to connect these 2 red nodes, well, I have to go to all the nodes in between. You see, as this network becomes 1 billion, I'll have to go all the way around. I can jump by 2, but still I will have to go. So it's going to scale as the size of the network. This is very bad for communication purpose. What about another type of graph there isn't? With the same number of edges, you can do something completely different, which is a random graph. Instead of making it very regular, you do it completely randomly. You connect one edge to the other, you just throw a dice to see how they're connected. In that case, you have short path lengths, right? If I take the same 2 nodes, I find that I can go like this, back and forth, and so the path is now 3 rather than the whole thing. And you can show that it scales, when it scales, it's much better. This graph has much shorter path length, but low clustering, because your friends don't know each other. Okay, they say, how do we go from one to the next one? What is the best of both worlds? And they say, let's start with the regular graph, and I want to keep the same number of edges. You can't cheat by saying everything is connected, right? You take this, you have fixed number of edges, and all you do is rewire. Take one of these connections and make a long-range connection. Okay, and that's exactly what they did. And they did, as you increase more and more, all of a sudden, and you can prove it mathematically thanks to these matrices. It's a problem of eigenvalues. If you've seen that in school, it's great. You can show that as soon as you rewire, you completely decrease the path length while keeping the high clustering. And so the path length is short again because you find long connection, just like my friend in Bogota. You know, that's a long-range connection. He knows people locally there. And it has both short path lengths and high clustering, the best of both worlds. And that's the small worldness properties, that the ability to have this close packing and long connection. It's called in neuroscience segregation, the high clustering, or localization, that you have process that takes place localized within the brain, whereas the long-range connection are called integration. You integrate different type of information. And the small world network have both segregation and integration. Another way to think about that is computation, this local computation, versus communication. And you need both. You need both to be able to do this small computation, extract feature of a visual input, and you have to be able to communicate that to other part of the brain. And that's what you have. Our scientist of the day who worked on that is Professor Danny Bassett. So Danny Bassett got a PhD in 2009 from Cambridge. We'll go back to a PhD work in a second. Then they went to UC Santa Barbara, where they did a postdoc there. And since 2011, they've been at University of Pennsylvania. They're still very young, but they've done foundational work on network neuroscience. They really consider as one of the leaders of the field. And for that, in 2014, they were given the MacArthur Fellow Award for the so-called Genius Grant. And they were the youngest person ever to receive it. They have by now over 470 scientific papers. They're extremely prolific. And they recently published a book called Curious Mind with a co-author who turned out also to be their twin brother, Perry Zerd. It's really worth reading, very interesting about creativity. So part of their PhD work was to extract information about small worldness property of brain network. And they did that with Ed Bullmore from Cambridge, very well-known neuroscientist. And they were able to show that the connectome has this small network property. And you have to realize that at the time, there was not so much data, connectome data that you can find online, MRI imaging. They really had to extract all the information while building the mathematical apparatus at the same time. And here is a representation or part of the paper, also another very influential paper, where they show that there are particular nodes within the connectome that have this long-range connection, and also these local properties of computation. And now you can understand a little bit better this matrix that I show you. We have this segregation, localization, all the activity on the diagonal are nodes that are close together, that are extremely well connected. But off the diagonal, once in a while, you have this long-distance connection that allows for integration of information. Okay? And now, thanks to their work and many refinements since then, we have a much better understanding of the overall structure of the brain that you can see as having this localized process and integration of different information coming together. And it was a long-standing debate a century ago whether the brain worked by sharing all the information, diffusion, or being completely localized. It turns out that you need to do both in order to do that. And this type of work by Danny and others was really foundational in the sense that it's a complete revolution for neuroscience. Two revolutions happened more or less at the same time. For one, the MRI data, the imaging data, starting to be good enough so that you can extract information. So you had experimental data becoming available. And on the other hand, work with Strogatz and Danny Bassett and many others built the mathematical apparatus to study this network. It's a complete revolution in the sense that now all these concepts that come from mathematics are completely routine in neuroscience. People do plenty of scan imaging techniques and all that. And they talk about centrality measure hubs and clubs and small worldness. These are becoming completely common in the normal language. They've integrated neuroscience, up to the point that there is now a very good journal published by MIT called Journal of Network Neuroscience that sole purpose is to study this type of system. So we have all these tools. What's missing from the picture? What's missing is that these two are very static. We talk about the structure, but not so much about brain activity, right? So there is a dynamic element that we are missing in this picture. And we can also probe that. There is another type of MRI that look at brain activity rather than the structure that's inside, not the axon, but really how much oxygen is being used at different parts of the brain. That's a good marker, a good proxy to tell you that the brain, certain part of the brain are excited. And you can look at two parts of the brain, and if they light up from the same stimulus, you show a picture of a square, and you see, oh, these two parts of the brain are all of a sudden active. Then you can say, well, there's somehow the two parts of the brain are correlated. There's activity in both of them. And so what you can do now is that, well, we can draw an edge between, even if we don't see how the signal is going. We see the activity going from one place to the other one. And you create another type of network, which is called the functional connectome, because it's related to the activity, that is different, but somewhat correlated to the structural network, because the information still has to go to it. So dynamic and activity is what we will talk in chapter 4 in my next Gresham lecture. And beside that, that's all, folks. Thank you very much.